The human eye is an amazing structure, even if it really has an imperfect sort of looking design. Normally, light comes in here through the pupil, must pass through this initial clear region, and then has to make it to the back of the eye, called the retina. And way here back in the back of the retina, past layers of cells, if the light can make it through all of these occluding structures, all of these things blocking its path, and it makes it to the very back, to these oddly shaped cells here called cone cells, then they'll actually be interpreted as the color that we see every day. Now, the visible light spectrum is interpreted by humans through the detection of three main wavelengths of light that correspond to three main colors: red, green, and blue. There's a special type of cell here in the back of the retina that's able to detect these three different colors, and it's called a cone cell. And there are three different kinds of cone cells that can recognize each of the different colors. Now, these three different types of cone cells can each detect a certain wavelength of light. Remember, light is actually energy, and it travels in the form of a wave, and the color spectrum essentially correlates to certain wavelengths that our eyes can see. The longer wavelengths are in the red spectrum, and the shorter wavelengths are in the blue spectrum, and the middle wavelengths that we can see are in the green spectrum. Each of these different types of cone cells are actually able to detect a specific wavelength of light. Together, these give you all the colors, all the combinations of all the colors that you can see. Now, this whole amazing process is possible because there are proteins like OPN1LW that absorb light at each of those different wavelengths. In the case of OPN1LW, it focuses on absorbing light from the red wavelengths. So if this is the cone cell membrane, right, we're really zoomed in here, and in the membrane of the cell, there are these proteins, specifically OPN1LW, that absorbs red light and essentially tells the cell and then ultimately the brain that, hey, this is red light. Now, in the case of the premature stop codon that we just talked about in the last exercise, the changing from a cytosine to a thymine causes a severely shortened protein that doesn't actually make it to the membrane. So now the cone cell can't detect the red wavelength of light and it just bounces away. So what I want you to do is think about this mutation and categorize it, whether you think it's a loss of function, a gain of function, or no change in function mutation.